Hello, everybody. Can you hear me okay? Good. Thanks to the chairs of the session and also, of course, Jay and Wolfram and really everybody who came and took time to travel from near and far. It's been really fun to talk to many of you and meet so many new folks. So today, yes, I'm going to talk to you about bioprinting of cardiogenic extracellular environments. But before I do, I just want to share, if I can get it to advance. It's hooked up to this. Oops. Oh, it does work. Okay, great. Thank you. My lab has had a long-standing interest in the interplay between the extracellular matrix, some 300 proteins in our body, and how they interact with stem cells, and especially stem cells that become cardiac or cardiovascular cell types. And then I want to talk about how this interplays with how we think about cardiac tissue engineering. So as most of you know, or I'm sure many of you, the extracellular matrix has many roles, not just as a structural entity. It can act as a barrier to therapy. It can act as a reservoir for growth factors. It also imposes some mechanical cues through mechanotransducers. And it's also a potent signal transducer. So it initiates many signaling cascades that have been well defined over the years, and most of them are related to more ECM synthesis, invasion, proliferation, and just general adhesion and movement of cells. But what we found a little more than 10 years ago is that the extracellular matrix is also a potent stimulator of differentiation. And we published in 2009 in 2D and then in 3D a few years later that if you provide just extracellular matrix proteins to stem cells, they can differentiate, and the differentiation outcome is dependent on the extracellular matrix protein that the stem cell was exposed to. And so I've actually presented this work, so I'm going to just summarize, but to bring us all up to the same page. At this same conference a few years ago, our efforts to derive a formulation, therefore, of extracellular matrix proteins that could drive specifically cardiomyocyte differentiation as a start. And so to begin to try to investigate that, we started to look at what proteins are actually present in the developing heart, especially during that critical period of proliferation to differentiation. And in the mouse, that's from about E12.5 to P2, or postnatal day 2. And so I'm just going to skim past this, but just to let you know that we did characterize 6 different ECM proteins, and we looked from the epicardium, I don't know if you can see this, yep, to the endocardium. We also looked at the interplay between multiple ECM proteins, and then we quantified that, and that really set the stage for an optimization experiment that was conducted by a postdoc in my lab at the time, Philip Jung. And what he did was to create an ECM array with 4 proteins that we narrowed it down to. He put different combinations into these 3D hydrogels, and we actually used polyethylene glycol to hold those ECM proteins in 3D so that they could be presented to stem cells that were entrapped. And we did different types of stem cells, but here, to get cardiomyocytes, we entrapped induced pluripotent stem cells. And the idea is that we could use a design of experiments approach to just sample a few elements of the parameter space, back out a theoretical optimum from the entire parameter space, and then test that experimentally by looking at CT&T expression, and then once we had our experimental optimum, confirm that with more functional studies. And this was several years of work that was sort of distilled down into one little graph. And actually he found what we call our OPT, or optimum formulation, of type 1 collagen, fibronectin, and laminin 111. And what he found is that in the absence of any other kind of soluble stimulators, like Wnt activators or inhibitors, you could routinely get about 40% cardiomyocyte specification. We used those same 3 ECM proteins, but in equal amounts, not according to the formulation. Did see specification, but statistically lower. So that means not only do the specific ECM proteins, but the relative amounts matter as well. The really cool thing about the formulation that we ultimately came up with is that it was strikingly similar to the composition of ECM proteins that we saw in the E16.5 embryos. And then even more fun, more recently, we've gotten a little more high-tech in our characterization of the extracellular matrix, and we've been using mass spectrometry instead of immunofluorescence or one by one. And Sophie Givens was able, in a study that we collaborated on with Sean Pelichek and Tim Camp, to look at ECM proteins expressed in fetal cardiac fibroblasts compared to adult. And actually those same 3 proteins or families of proteins came out as the most differentially expressed between those 2 types of fibroblasts. So that's pretty interesting too. Fibronectin, type 1 and 3 collagen, and laminins, including 111. So that's previous work, and now what I want to share with you is some of the work we've done since then, both on the mechanism side of things and then also on the applied side of things. So the sort of bottom line that we found is that we're sort of interested in how is it that ECM engagement can drive specification. And we started to look at the Wnt signaling pathway and also the focal adhesion complex. And we wondered if there was an interplay, a direct interplay between these 2 complexes of proteins inside the cell. And we found a couple of papers, some of which linked ILK, which is integrin link kinase, which is part of the focal adhesion complex, to GSK3 beta, and that these 2 proteins actually co-immunoprecipitate, and ILK can block phosphorylation of GSK3 beta. And so with that hypothesis, we first looked to see what did beta-catenin activation look like when we expose iPS cells to our optimized formulation. And what we saw is that by day 4, we had a significant increase in beta-catenin localized to the nucleus, which started to support that hypothesis. Similarly, we saw an increase in phospho-ILK. We've done many more experiments since then that sort of further confirm this interplay between elements of the focal adhesion complex with Wnt signaling. This work was done by Sacha Robert. I point you to this recent publication so you can see all the details. But essentially what he found is that not only was ILK involved, so if you inhibit with CPD22, you see a reduction in that differentiation we saw with OPT. Similar with inhibition of focal adhesion kinase, we see an even more dramatic decrease, further supporting that link between focal adhesion complex and Wnt signaling. We're further exploring this in the context of cardiomyocyte specification, but we have also started to look at, I'm just going to skip over this for a second, endothelial specification, and whether or not ECM proteins can drive endothelial specification without additional stimuli. And so in this case, we looked at a different set of proteins, again, fibronectin, but also type 1 collagen, laminins 111, 411, 511, and then we used a matrigel, or the M that you see here, control. And what we found is that without any small molecule Wnt agonists, we see endothelial specification. This is CD31 staining, only in the laminin 411 condition, or at least the only one that's statistically significant. If we add in also the small molecule agonist, we see it in more of these conditions, but most prominently again in 411, or laminin 411. So Michaela Hall, who was looking at this, did a similar set of experiments as we did with the cardiomyocyte specification. She looked in the context of laminin 411 this time, not in the cardiomyocyte OPT formulation. If we block ILK and or FAK, do we see a decrease in endothelial specification? And indeed, she did see that compared to the controls. So you can see this is our 411 condition, DMSO, and then with the inhibitors. She also saw a complete ablation in the presence of IWP2, or Wnt antagonist. And so what she found is something similar. We think that ILK and FAK are interacting with elements of the Wnt signaling pathway. She did further work that's been published in Stem Cell Reports, noting that the intracellular domain of Notch also seems to be involved and can be blocked, similarly blocked to inhibit arterial gene specification, which is interesting. We also utilized IPS line that had a stabilized beta-catenin in the presence of DOX. And what you saw is that C indicates collagen, so collagen with DOX, you'd see very little CD31 specification. 411 without DOX, so this is without DOX, you see a CD31 specification as we did in our other experiments. And if you add DOX to the type 1 collagen condition, you actually start to see CD31, but you don't see any change if you add DOX to the 411 condition. Further supporting the fact that ECM engagement somehow is eliciting beta-catenin activation and movement into the nucleus. This is just a little teaser for the end. She has gone beyond just individual ECM proteins in driving endothelial specification and has started to implement the same sort of protocol that Philip Jung did to find an optimized ECM formulation, so beyond 411. I don't have all of the details of those ECM proteins here. This hopefully will be coming soon. But something interesting to note, there is laminin 411 in these formulations. Interestingly, if you add another layer of ECM to the top, or if you add VEGF, you see even more specification. And in the presence of SB, so inhibitor of TGF-beta, you see a nice monolayer that forms. In the absence of that, you see contraction, compaction of the cells in the area in more tube-like structures that form. So we think there's an interplay between other mesenchymal cell types and the endothelium in the presence or absence, depending on how you look at it, of a TGF-beta inhibitor. So those are some interesting studies coming out and just kind of further emphasize the idea of generating a formulation of ECM proteins that can drive specification. And you might think, well, we have lots of other soluble molecules that can do that much more efficiently. What's the benefit of using the extracellular matrix? And for us, it's, well, it's interesting, first of all, but second of all, it could be very powerful in spatially patterning differentiated cell types without worrying about the control of molecules that might diffuse in whatever setting you're thinking about. So that's kind of the mechanistic piece and where we're going with that, and try to understand how the ECM does drive differentiation of stem cells. And now into some opportunities to apply our understanding of that cell ECM engagement. So I have lots of interest in my lab in 3D bioprinting. It seems like all graduate students are very interested in that concept. And actually in our lab, it kind of makes sense because our ECM formulations lend themselves very well to a bioink that you might consider 3D printing. And so that together with some interest in some industrial collaborations that we have with some of our local medical device companies, are interested in human model systems of cardiac tissue. And more than just a strip of tissue, they're really interested in something that we could measure pressure volume dynamics and impose volumetric flow, and ultimately to evaluate clinical grade devices, medical devices. So it has to be fairly large and ideally chambered in a way that can be perfused with fluid. So it was a tall order. We tried to think of ways that we could address this, and we immediately came upon several problems. The first problem is that the ECM protein mixtures that lend themselves best to cell viability and ultimately differentiation are very low viscosity. We tried to add different kind of fillers and other things, and the only thing that we could really land on was gelatin methacrylate, which still enabled the same cellular processes that we were interested in, but still very low viscosity. So we looked to some work coming out of Adam Feinberg's lab at Carnegie Mellon University, who was using slurry-based 3D printing, and we decided to use that. And so what we did is we took an MRI scan of a human heart, we scaled it down to the size of a mouse heart, and we simplified it heavily to have just a single inlet and outlet flow port. I'm not hoping this is going to work. Not sure. Oh, here we go. So this is just sped up, and it's actually upside down, so the vessels are at the bottom, moving up toward the apex at the top. And again, we're using gelatin methacrylate plus our optimized ECM formulation for cardiomyocyte specification. We initially tried to print with cardiomyocytes because we can generate many in the lab. We know that there's been some success doing that, but it takes a lot of cardiomyocytes. And we find that just because of their low proliferative capacity and a limited ability to move, that we had a hard time getting much contiguous muscle function in this setting. And so instead, because of that low cell density, we decided to do something different that we term in situ differentiation. So we're printing with stem cells in this case, allowing them to expand, and then differentiating them to cardiomyocytes. And this was sort of practically useful for us, but as we look in hindsight, it's a little more akin to development, sort of like an organoid where you get that expansion of the stem cells first and then specification. That way we also don't need to disrupt the cell-cell connections or the cell matrix connections to make the printed structure. This is a table you can't read. No, you can read pretty well. But if you want to check out the details more, you can look in the manuscript. But essentially, this was our very small optimization profile to figure out how to integrate the gelatin methacrylate with our OPT formulation to get the best expansion of the stem cells and then the best differentiation after expansion. And so just a few attributes of the structure. So now we were able to, I'm going to get this eventually. Oh, well, at least you can see it. So you can see that the structure can be perfused. We can hook up tubing. There's no leaking. This is what a cross-section of the template, the digital template looks like. Here's our print. It's actually pretty low resolution compared to some other things we printed, so it's about 300 to 400 micron resolution, and the walls are about 1 millimeter thick. And this is what the structure looks like ultimately. This is the overall fidelity. And what we found in these structures is really nice muscle formation, actually. And Jay Zhang's lab did this immunohistochemistry for us for troponin I, smooth muscle actin, and CD31. And basically, this comprises nearly all the nuclei in this structure, majority of which are cardiomyocytes. I hope I can get this over. I don't know why. If I have to double click or single click. Anyway. All right, I'll just tell you what this one does. It's just too bad. This is one of our structures, and you can see it, the entire structure beating synchronously. You can see this in macro scale in the dish. And we can make these routinely. The longest axis is about 1.5 centimeters. So in our terms of what we usually work with, this is fairly large for us. This is a cross-section of a chamber, and you can see the troponin T positive cells along the exterior surface in a pretty nice contiguous muscle sheet. In the interior, we drew this line so you could see where the scaffold actually ends. There are no cells, and this is primarily because we didn't perfuse this initially. We can perfuse it, but we didn't have a bioreactor in the very beginning, so we just had it in the incubator with convective movement of media. But we did get a fairly thick muscle layer, up to about 500 microns in thickness, and you can see some of the, if you zoom in, some of the striated muscle. It's not well organized, I would say, right now, and I'll show you in the end of the talk. We have now developed a bioreactor and think that with imposed volumetric pressure, we'll get more alignment, but I'm also been very interested to watch amongst the talks today and yesterday, the different modalities for alignment, and maybe things that we can impose on this structure. This is more staining just to show the relative maturation of the structures, and actually we've kept some of them going in the lab for more than 2 years now. So they beat robustly and they don't slow down. I really hope I can get the optical mapping to work because it really conveys the level of muscularization that we get here. So, oh, it did it. No? If anybody knows how to help me, you can go. Click on move. Just the left corner? Okay. Okay. So with this one, you can see the, this is a voltage sensitive dye, and you can see it propagating, I hope you could see, around the edge of the structure. And you can see that movie corresponds to this top row, this activation time map, APD80. You can see this dark structure, or dark mass in the middle, sort of indicating maybe there's no cardiac muscle there, so not a very complete muscle layer. But it was interesting because we actually could see cardiomyocytes there. So what we did is we decided to electrically pace it. So we took another one where we still had sort of these gapped regions. This is the spontaneous activity, and then this is what it looks like when you pace from the other side. Oops. So that's a spontaneous activity. And then with pacing from the other side, you get pretty robust, nice, consistent muscle layer. Again, not well organized yet, but in our experience, this is pretty big achievement just to have the contiguous muscle. Then we also used a pressure transducer with a conductance catheter to get an idea of our pressure that we could build inside the structure. So these are just very rudimentary pressure volume dynamics. And you can back out a pressure volume loop. You can see that there are no valves, so there aren't nice corners, but in the presence of different drugs, you can see the change in that stroke volume as you would expect. So this is just the beginning, very small pressures being built, but again, the capacity to register those and enough muscle mass to detect those pressure changes. What we're doing right now is collaborating with the Center for Engineering Complex Tissues at the University of Maryland to develop a bioreactor. And we have inlet and outlet lines to which we're adding valves that act as a kind of quasi-mitral and aortic valve. And the idea is to actuate them to mimic a native cardiac cycle, and then add electrical stimulation to also have it beat at the proper time. We think, well, we know already that with perfusion, we're going to get a much thicker muscle mass. So already just looking at the expansion of stem cells in a perfused versus static culture, these individual cells won't go on to proliferate. Eventually they'll be lost, and we'll just end up with that layer on the outside that you saw before. Here we're seeing big colonies right away that will be differentiated into muscle. We modeled what we think our stroke volume will be if we're able to get that full thickness muscle. So right now we have that exterior layer. We can move about 5 microliters of fluid. If we model in a single, the force of a single cardiomyocyte and the number of cardiomyocytes we think we have in this structure, we think that with the full thickness or even just the inner wall thickness, we'll be able to move closer to 10 to 12 microliters, which is approaching the native volume. So with that, I just want to share some of our future directions where we hope to take this model and just our understanding of cell ECM interactions. We're looking at modeling cardiac diseases, especially those where progression is accelerated by dynamic volumetric loading, like hypertrophic cardiomyopathies. We're also in the process of imparting an epicardium and integrating epicardial-derived cells. We'd like to, in the future, impart an endocardium and quasi-vasculature, kind of building off of Michaela's work. We're also in the process of grafting this in a rodent model in the heterotopic position. And then just taking this talk full circle, we are also imposing medical device interventions, and maybe in subsequent years, I can show you some of the outcomes of those studies, both ablation and cardiac pacing in this context. So with that, I've mentioned all the lab members as I talked about their work, but also Gary Lyons, Paul Campanola from the University of Wisconsin, Jay Zhang from University of Alabama, Birmingham, Dwayne Townsend, Lena Telkachova, Angela Mortari, Michael Kaiba from the University of Minnesota, and Sean Pelichek from Wisconsin. With that, thank you all very much.